Now in each of these steps you could have intermediate species like S3O6 2-. There could be a bunch of these things, these sulfur intermediates that are being produced. So these can then become source for other microorganisms to do it. So what a sulfate reducing microorganism will do is it will take the sulfate and just maybe produce this intermediate. Then there will be another family of microorganisms who will be present, who will take this and take it to the next step. Then there will be some other microorganisms who will be present, who will take it to the next step and next step. So there could be a whole complicated family of microorganisms who are present when you have these sulfate things. And these things are quite common. Where do you think these will be common? Where do you have a lot of sulfur, sulfate business going on? Hydrothermal deposits. So there, this is the process that could be happening. So all these oxidized things can just accept the electrons and produce their reduced forms, and this is a form of anaerobic respiration. Okay? Now, this is very simplistically, but now let's try to understand a little bit about aerobic respiration. Okay? Now, there will be primarily 4 stages of aerobic respirations. So let's start from here. The first step is glycolysis. Now, so what was respiration? Getting that sugar and converting into CO2, right? So this is what will happen inside your cell to do what? To produce energy. That's what we are doing here. And what is your electron acceptor right now? Oxygen, right? So keeping that in mind, we will see what are the different stages. So the first step is glycolysis. What do you think the meaning of glycolysis is? What is glyco? Sugar. And lysis? Splitting. So this is splitting of sugar, right? So you can call that sugar splitting. Now, we are calling it sugar splitting, but we eat a lot of different, we don't only eat simple sugar. We eat complicated sugars. Now those complicated sugars need to be broken down into glucose. So that is another step. You can think of it as the pre-planning step, in which you say you have starch in your body. That starch is what? It's a polymer of the glucose, right? Or fructose and things like that. Actually, it's the alpha, if you remember, the 1,4, the glycosidic linkages, how we were doing, right? Anyway, so you have a polymer. All these glucose molecules are there. So the first step will be to split those glucose molecules. So like take water, split it, take water and split it. So you will start getting individual glucose molecules, right? This process is starting when you have those individual molecules. So you can think that there is a pre-planning stage that is also going on, right? Now, sugar is not the only thing that we eat. We also eat a lot of proteins, right? So what happens to that? We'll see that when there is a particular molecule that is being produced in aerobic respiration that is common to the glucose, the proteins and things like that. So let's just start simple, start thinking about the sugar, just the sugar. Okay? The location of this is the cytoplasm of the cell. So you have the cell, you have the nucleus, you have the other cell organelles. This white part, that liquidy part that we have, the gel-like part, there is where the cytoplasm happened. It doesn't happen inside the nucleus, inside the mitochondria, or inside any other cell organelle. It is happening in the cytoplasm of the cell. Why this is important? Because this produces something, a pyruvate, and we'll see that gets into the mitochondria, and that is why we call the mitochondria the powerhouse of the cell. Because there will be 4 steps. One of the steps is this, but this is the only step that happens outside the mitochondria, not inside the mitochondria. Okay? Now, what is the process? The process is a glucose molecule, and how many carbons are there? 6. So think of this as a C6, is split into 2 pyruvate molecules. And the pyruvate is a C3. Okay? This process does not require O2. So, if we are doing anaerobic respiration, this process, the glycolysis, is kind of like common. Or anaerobic respiration could also have a thing called fermentation. Have you heard about fermentation before? In what context? Alcohol making is one of the processes. Inside our body, we also have fermentation. When will we have fermentation? Just think, when will we have fermentation? Anaerobic condition. When we do not have a lot of oxygen, then we tend to start fermenting. Now that fermentation, the alcohol is one type of fermentation. So that is the alcohol fermentation. But you might have heard lactic acid fermentations. Have you heard that when you're running or when you're fatigued, your body aches, right? That ache is because of that lactic acid formation. Just imagine that when you have a lot of oxygen, that oxygen will help you to break down glucose fully and release a lot of energy. But when you do not have a lot of oxygen, after a while, you will not be able to produce a lot of energy. So, when you run, like long distance runners, the idea is to not, so you can run 2 ways. One, in which you can run controlled, in which your breathing is normal, and you can take in a lot of oxygen. So while you are running, that is called aerobic running or aerobic phase. Why? Because so you have plenty of oxygen to break down that sugar and release the energy. Break down the sugar and release the energy. But sometimes when you run fast, say you ran fast for like 10 minutes, then you start to build up that anaerobic condition in your body, because when you're running fast, you cannot breathe that. So essentially you're cutting down the oxygen supply. So when you're cutting down the oxygen supply, the sugar is not able to break down properly. A type of fermentation starts happening in your body. That's why you get tired. That's why you cannot run fast. You get cramps. Because A, you're not getting energy, and that lactic acid is getting stored in your body. Okay, we'll look into that. But this is the process that is common for both anaerobic and aerobic respiration, because you can see that this process does not require any oxygen. Okay, we'll take a look at what those processes are. Now, the energy output of this. So all these 4 processes will have some energy requirement and some energy output, right? It's like think of it as an activation energy and the exothermic processes, right? In here, it takes 2 ATPs to start the process. But it produces 4 ATPs. So there is a net gain of 2 ATPs. Plus, there is an additional thing that is getting produced that also can store energy, but not ATP. It's called NADH. It's a molecule, and we get 2 NADH. So we understand ATP as a seat of energy. There are other seats of energy. There are other currencies of energy as well. NADH is also another currency of energy. Okay? So in here, it's maybe, definitely it is different than the ATPs, but this is also, think of this as a battery that is getting charged, right? So net production is 2 ATPs and 2 NADH batteries, okay? Now, let's look into the glycolysis in a little bit more detail. I'll first draw a simplistic version of this and then go into the more detailed. In glycolysis, so you start with your food. So you have your food. Now this food could be anything, not necessarily just sugar, right? So this could be your normal sugar. You can also have fat or lipids. You can also have proteins or the amino acids. Okay. Now, sugar and all these other kinds of glucose. Now, in glycolysis, what will happen is, this is the glycolysis part that will happen in here. So this is glycolysis, and you start producing a pyruvate, as I mentioned here. You start producing pyruvate. Okay. Amino acids also produce pyruvate. Okay. This pyruvate has to then get converted into a thing called acetyl CoA. Just imagine that you have an acetyl group, and then there is this enzyme which is called the CoA. Okay? These two come together, form like a bond, like we saw in many other things, like two things coming together and forming a bond. So you are forming acetyl CoA from this pyruvate. Now, the fats can directly produce the acetyl CoA. Even the amino acid can actually directly produce the acetyl CoA. This process is the only process that is called glycolysis. This is called beta oxidation. We'll not get into that. And this is amino acid catabolism. These are just names, but essentially you understand that everything is coming down to these simplistic molecules. And you can see that the breakdown has already started to happen. So you had this 6 sugar, you started to get the pyruvate, which is a 3 carbon. And then from this pyruvate, we get an association acetyl CoA. And this acetyl CoA becomes the fodder for another step in the aerobic respiration called the Kreb cycle, but we'll get into that. So in this glycolysis, sugar to pyruvate, let's dig a little bit deeper just for your, I'm not expecting you all to memorize this, but just know these things, at least have it in your notes so that you can say, hey, we kind of know what glycolysis is. So let's see glycolysis in cytoplasm. So we are still in the cytoplasm. We are trying to see what happens. So you have the glucose, which is a C6 molecule. This gets converted into glucose 6 phosphate. What is your intuition? What does your intuition tell? Well, where is that phosphate coming from? It's coming from ATP. Now in here, you can see that an ATP is converted to an ADP. So there is one. We saw that it is taking 2 ATPs to start. We see this process starting in here. So in here, to convert one glucose to the glucose 6 phosphate, you already took one of the phosphates. So now this becomes a C6P. This is just a way to write it. The second step is you will have, this becomes a fructose 6 phosphate. Glucose and fructose are what? They are isomers. So they just one isomer is getting converted to the other. And then this gets converted to 2 molecules of again, you can see an ATP gets converted to an ADP. In here, you get minus 2 ATP. This is the energy consuming part of the molecule. Okay. Now, from here, you start generating other types of energies. The first type of energy production is the production of NADH that we saw in here. In here, what happens is you produce a compound called 1,3-biphosphoglycerate, and you produce Na DH. And yeah, so this is also 2. From this molecule, you then produce again 2 molecules of 3-phosphoglycerate PC3, and what is happening in here? You see a biphosphoglycerate, and now you only see a phosphoglycerate. What happened here?
Phosphorus
was released. That phosphate group was released. What do you think happened to that phosphate group? Now in here, it starts to produce the ATP. How many ATPs at this step? Why? Because you can see that there are 2 of these things happening. So you will start getting 2. How did you get this? From the ADPs. from this, you get hydrolysis. So from the previous step, you interact with water. This 3-phosphoglycerate interacts with water, and then you get 2 molecules of phosphoenol pyruvate. This is also a phosphate and a C3. What do you think will happen again? Think. We showed that it produces 4 ATP. We see that 2 ATPs are produced here. What could happen here? So we see this phospho group in here. What could happen? This phospho group can be released again. Because essentially, we are trying to make sense of all this, because you need to produce 2 ATPs. We have 2 molecules of this. If we release this phosphate group, and an ADP takes that, then you again generate 2 ATPs. So exactly that happens. So this becomes a pyruvate, and 2 ATPs are produced from 2 ADPs. This is the end of the glycolysis cycle. Any doubts with this glycolysis part? Okay. If this is clear, then let's move to the next step, which is this. So pyruvate to the acetyl-CoA. So the next step is the conversion of the pyruvate to the acetyl-CoA. This might not mean much to you, but just know these names, because these names are important. Pyruvate, acetyl-CoA, they will come over and over again, right? And if you think about biochemistry, just knowing these names is important. These individual steps, not so much. Glucose to pyruvate is called glycolysis, because essentially you are broken down a 6-membered glucose into 2 3-membered pyruvates, right? So step 2 is pyruvate oxidation, in which this happens when you are entering the mitochondria. So glycolysis happened in the cytoplasm. Now we are entering the mitochondria, and this is where the pyruvate oxidation happens. What happens in here is the 2 pyruvates that enter, a carbon atom is removed from the pyruvate. To form what? What do you think the carbon was removed? So you have a 3-carbon pyruvate, and you removed that carbon. So what do you think that carbon would have become? Each of these will become carbon dioxide. So you start getting the carbon dioxide out in here. Now the remaining 2 carbon molecules, so you have a C2 molecule. Now this C2 molecule is attached to an enzyme called CoA, and you form acetyl CoA. This becomes important, is acetyl CoA, because now this will become the energy. We will break down this acetyl CoA to release more energy, right, in here. In here, you produce 2 NADH again. There are some electrons, high energy electrons, that converts the NAD plus to NADH. So you are seeing that in step 1 and 2, we were not able to produce a lot of energy yet. 2 ATPs there, 2 NADH, and here 2 NADH. The real action will start in step 4. But the step 3 becomes very interesting, because this is a cyclic process that happens inside the mitochondria. So now you are starting to enter inside the mitochondria. Till now we were outside the mitochondria. Okay, so now the step 3. So now we are here. All these things were happening in the cytoplasm or in the process of entering the mitochondria. Now we are in the mitochondria. Now let's see what happens in the mitochondria. The first or the third step that happens in the mitochondria is called the Kreb cycle. What is the other name of Kreb cycle? The TCA. Okay. Now there will be citric acids in here, and that is why we can call it the citric acid cycle as well. Citric acids are carboxylic acids in themselves, right? And we can also call them tricarboxylic acid cycle. So there are various names, but they all refer to the same thing. Krebs cycle, tricarboxylic acid cycle, or the citric acid cycle. All these are referring to the same thing. In the citric acid cycle, so what is the location? The mitochondria. What we call the powerhouse of the cell. Okay. Now we have the acetyl CoA in the process, and this the other thing is this is a cyclic process. Now the acetyl CoA that enters here, you can think of this as it is fully burnt or completely oxidized. Okay? To get CO2 and think of it as like the CO2. So this acetyl CoA, which was made up of the pyruvate plus the CoA enzyme to give an acetyl CoA, this now is getting fully burnt in a cyclic process called the Krebs cycle to release the waste product of respiration, which is the carbon dioxide. Now, there is a lot of electrons that are produced in the process in here. In here, you get a final production of 2 ATPs in the Krebs cycle. The electrons help you to produce 6 NADH, which is the battery that we were producing. And you also get another kind of a battery called the FADH. Till now, we have not seen